Hello guys, welcome to my YouTube channel. So in this video, we are going to learn about glycolysis. So I'm going to show you where glycolysis occurs in our body, when does it occur, why does it occur, and some of the organs that are involved in glycolysis. Now, let's see. I'm going to use this to represent the cell. So, what is glycolysis in the first place? So glycolysis is the process by which the body breaks down glucose molecules into other precursors that can be taken into other pathways for the well-being of the body. Now, glycolysis mainly occur when there is excess glucose in the circulation. For example, after eating something like a carbohydrate, any meal that's going to increase the level of glucose in your body. So when there is excess glucose inside the circulation, insulin hormone is going to activate the hexokinase or the glucokinase with respect to the cell that's going to be involved. Now you know glucokinase is mostly involved in mostly found in the liver. So when it is in the case of the liver, we're going to talk of glucokinase. When it is in the case of other cells, we're going to talk of hexokinase. Now, when there is excess glucose, so this is going to represent our circulation. Then this is going to represent the cell. Now, when there is excess glucose inside the circulation, so I'm going to present glucose as 6 carbon 3, 4, 5, 6. So this is glucose inside the circulation. Now, I said when there is excess glucose inside the circulation, glucose is going to get into the cell. So by the blood receptors. So these are called blood receptors. Blood receptors. So guys, see this. The blood receptors are named based on the cell. For example, we can have blood 1, blood 2, blood 3, blood 4. So which is blood 1? Blood 1, that one we are talking of the mostly the brain cells. So brain cells are blood 1. When it is blood 2, mostly refer to the liver cells. So the erythrocytes are blood 2. So liver cells. Blood 3 are mostly present in the kidney cells. In the kidney cells. Then you have blood 4, mostly present in the muscle cells. Muscle. Now, these are blood receptors for glucose molecules. So they're named based on the organ. So in this case, let's just talk of a liver cell. Let's just say this glycolysis is going to happen in the liver cell. So I'm going to use blood 2. Alright. Now, glucose will get into the cell by blood receptors. So it's going to get into the cell. So we have the 6 glucose, the 6 carbon glucose. Now, when it gets into the cell, immediately it's acted upon by an enzyme that was activated by the insulin hormone. Now, what is the name of this enzyme? This enzyme is going to be called glucokinase. Glucokinase. But don't worry, in other books you may see it written hexokinase. So it just depends on the cell you're working with. Now, I said you're going to use a liver cell, so specifically you're going to have glucokinase. When it is all the other cells, hexokinase. Now, so glucokinase, so you see it's written kinase. What comes in your mind when you hear the term kinase? Kinase is going to phosphorylate. It's a phosphorylator. So it's going to add a phosphate group. So when it adds a phosphate group onto this glucose molecule, it's going to become glucose 6 phosphate. So it's going to add a phosphate group at the 6th position. Now it's going to attach a phosphate group there. Now this is called glucose 6 phosphate. Alright. Now it adds a phosphate group. So where does it get that phosphate group? It's going to get it from ATP. So you're going to use a molecule of ATP here. So we're going to have ATP going to be reduced to ADP, and this ADP. Alright. Now that's glucokinase. So we have glucose 6 phosphate. So remember, we phosphorylate this glucose molecule primarily so that it doesn't get out of the cell, so that it can stick inside the cell, so that it can undergo glycolysis. Because if you don't phosphorylate it, to me, it does have the capability to get back to the circulation through the receptors, and that's what we don't want. We want to make sure that we break down this glucose molecule. Now, glucose 6-phosphate is going to be acted upon by another enzyme. So, we are going to form another 6 carbon, 3, 4, 5, 6. Then it still has a phosphate group at the 6th position. So, what is the name of this molecule? This molecule is called fructose 6-phosphate. So, fructose 6-phosphate. Alright. Now, we have moved from glucose 6-phosphate to fructose 6-phosphate. The phosphate group is still at the same position, the 6th position. So what we have done is that we've just shuffled the position of those particular atoms so that we have the fructose 6-phosphate. So what is the name of this enzyme? This enzyme is going to be called phosphoglucose isomerase. Phosphoglucose isomerase is just going to change the position of those particular atoms. So to form fructose 6-phosphate. Now let's get down. Fructose 6-phosphate is going to be acted upon by another enzyme to form fructose. So we're going to still have 6, 3, 4, 5, 6. From fructose 1,6-bisphosphatase. Yep. Fructose 1,6-bisphosphatase. Now, some of you may wonder why do you say bisphosphatase? So I said when there is a distance between the functional groups, the 6. Okay, you see, it is a phosphate group is at position 1. You can see a position 1 and at position 6. Now, when there is some distance between these two, we say bis. But when there is no distance, for example, if it could be at position 1 and position 2, we could have just say bi. So in this case, we are going to say bisphosphate. So don't be confused or surprised. Now, we've added another phosphate group. Remember, phosphate group was at position 6. We've added another phosphate group. So we're going to have a kinase. So what's the name of this enzyme? This is going to be called phosphofructokinase. Alright. This is an enzyme, another enzyme. It's a kinase. To me, it's going to phosphorylate. So it's going to add a phosphate group onto fructose 6-phosphate to give us fructose 1,6-bisphosphate. Now, we're going to get that phosphate group from ATP to give us ADP. Alright. Let's get up. Fructose 1,6-bisphosphate is going to be cleaved. It's going to be cleaved into 2. Now, what's the name of this enzyme? Aldolase. So we can have aldolase enzyme. This enzyme is going to cleave fructose 1,6-bisphosphate into dihydroxyacetone phosphate. I'm just going to abbreviate it as DHAP with it's 3 carbon. Alright. Then, and glyceraldehyde 3-phosphate. So, glyceraldehyde 3-phosphate also has 3. Alright. Now, we want to move down. The hydroxyacetone phosphate cannot go the remaining steps. So what are you going to do? You're going to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So we're going to, this junction here is reversible. So we're going to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So what's the name of that enzyme? This enzyme is called triose phosphate isomerase. Now, triose phosphate isomerase is going to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. That is to tell me that from glyceraldehyde 3-phosphate going down, everything is going to occur twice because glyceraldehyde 3-phosphate itself is capable of undergoing the remaining steps. We've added another 3-carbon molecule, the dihydroxyacetone phosphate. So to me, everything here is going to occur twice. So mark that. You're going to see how it is going to help us. Now, glyceraldehyde 3-phosphate can then be converted into 1,3-bisphosphoglycerate. Alright, this is B. 1,3 is phosphoglycerate. Now see this. We only have one phosphate group, but now here we have two. I mean, we have phosphorylated again. Now, but in this case, you're going to see what's going to happen. Something very fine. Now, the name of the enzyme is called glyceraldehyde 3-phosphate dehydrogenase. Now, mark this, guys. Whenever you hear the term dehydrogenase, will always mean that there's always the formation of NADH or one of those reducing agents or electron carriers. Now, in the process of this enzyme, we are going to convert NAD plus into NADH. It is a dehydrogenase. So to mean it's going to rip off the electron from glyceraldehyde phosphate, add it to NADH so that it give us NAD plus to give us NADH. Yes. That's called the nicotinamide, the nucleotide. Now, correct. Now, here we are not going to use any molecule of ATP. So what is going to happen? This enzyme here is going to add a phosphate group, an organic phosphate. It has that property. It's capable of adding a phosphate group. So that it's going to add a phosphate group at position 3. So that you are going to have 1, 2, 3. So at position 3 and at position 1, you have a phosphate group. Alright. Now, let's go down. From 1,3-bisphosphoglycerate, we are going to have another enzyme working on this one. So that we can now have a 3-phosphoglycerate. Right, we've gotten rid of that phosphate group. Now, what's the name of this enzyme? This enzyme is going to be called phosphoglycerate kinase. It is a kinase. It's going to phosphorylate. Now, what is it going to phosphorylate? ADP. It's going to add a phosphate group onto ADP. Now, we're going to have ADP. It's going to be phosphorylated to give us ATP. But mark this thing, guys. I told you from glyceraldehyde 3-phosphate going down, everything is going to occur twice because we converted dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So everything here is going to occur twice. So that therefore this NADH, you are going to have 2 molecules of that NADH. This ATP, you are going to have 2 molecules. ADP, 2 molecules. You are going to see how that is going to help us. 3-phosphoglycerate is going to be acted upon by another enzyme. So, 1, 2, 3. Alright. 3-phosphoglycerate is going to be acted upon. Of course, you know it is at position 3. To give us 2-phosphoglycerate. Now, 3-phosphoglycerate is going to be acted upon by an enzyme called phosphoglycerate mutase. Phosphoglycerate mutase, right? Phosphoglycerate mutase is going to act on 3-phosphoglycerate to give us 2-phosphoglycerate. So it means it's just going to change the position of the phosphate group. So we're going to have 1, 2, 3. So the phosphate group is at position 2. Now, alright, let's move down. 2-phosphoglycerate is going to be acted upon by another enzyme to give us PEP or phosphoenol pyruvate. It's going to be called phosphoenol pyruvate. Now, what's the name of that enzyme? That enzyme is going to be called enolase. Enolase enzyme is going to act on 2-phosphoglycerate to give us phosphoenol pyruvate, or you can just see it in some books, it's abbreviated as PEP. Phosphoenol pyruvate is going to be acted upon by another enzyme. This enzyme is called pyruvate kinase. Pyruvate kinase. See this is a kinase. It means it's going to phosphorylate. So what is it going to phosphorylate? ADP. So we're going to have another molecule of ATP being generated here from ADP. And remember, I told you guys everything here occurs twice from glyceride phosphate. So you have two molecules of each. Alright, pyruvate kinase is going to act on phosphoenol pyruvate to give us pyruvate. And guys, this is the last product in glycolysis. Now let's see. Pyruvate kinase has said phosphoenol pyruvate to pyruvate. Now, what's the fate of this pyruvate? Because the fate of, the aim of glycolysis is just to give us precursors for other pathways that will be needed to generation for the generation of ATP. Now, pyruvate has three fates or can have three pathways to go through. For example, it can go to lactic acid pathway or lactate. You can move to TCA or the tricarboxylic acid cycle, or also called the Krebs cycle. It can also move to alcoholic fermentation. Alright. Now, the fate of pyruvate is determined by whether there is oxygen or presence of oxygen or absence, right? Or absence. Now, when there is oxygen, to mean there is anaerobic respiration. So this anaerobic is going to go to TCA or the Krebs cycle. When there is anaerobic lack of oxygen, it can move to either alcoholic fermentation or lactic acid fermentation. Now, there's something I want to show you this. Look at this one. When pyruvate is going to be converted into lactic acid, it's going to be all lactate. It's going to be converted to lactate. Now, there's going to be an enzyme that's going to come to play. Pyruvate is going to be acted upon by an enzyme called lactate dehydrogenase. To mean what it's going to do, it's going to take up the NADH that we generated from glycolysis. So we're going to have NADH. It's going to be converted to NAD plus. Nicotinamide dinucleotide. It's going to take those electrons, add it to pyruvate, we generate lactate. Lactate can then be taken into the liver, right? To generate ATP or other pathways. Alcoholic fermentation. Pyruvate can still be acted upon by another enzyme so that we can get into alcoholic fermentation. Now let's see. Pyruvate can be acted upon by an enzyme that's going to give us alcohol. So in this case, this enzyme is called decarboxylase. This is going to be pyruvate decarboxylase. Pyruvate decarboxylase is going to act on pyruvate to convert it to alcohol, but before it does, going to convert it to acetaldehyde, which can then be converted into ethanol. So this can be catalyzed by another enzyme that is called alcohol BH. Now, TCA also, when there is oxygen, you can go through. There's a lot of things we are going to discuss in another video, but I want to see that. So this is all that you have in glycolysis, guys. But let me show you something. We're going to have the products of glycolysis. So what do you get? Number one, you obtain or generated two molecules of NADH. So two NADH. What else did we generate, guys? We generated two molecules of ATP, two molecules of ATP, that is 4 ATP. So gross ATP. Gross ATP, we are going to have 4 ATP. But how many did we use, guys? Remember, when we started glycolysis, we used two molecules of ATP. Here and here. Because remember, before we did this one, glyceride 3-phosphate, everything here occurred once. So we're going to, what have we used? We used 2 ATP. So what is net ATP? Net ATP is going to be 2. So we generate 2 molecules of ATP from glycolysis, and the generation of ATP through this process is called substrate level phosphorylation. Now, we use generated pyruvate, which is a precursor for alcoholic fermentation, we have lactic acid fermentation, and also for Krebs cycle. So in another video, we are going to see how we can, the pyruvate we generated here, we are going to use it for glycolysis. We are going to run the glycolytic pathway so that we generate other precursors for the electron transport chain. The main aim for this metabolism is just to generate the energy in form of ATP for the body, right? Now guys, think you're done. That is all about glycolysis. You have glucose molecule acted by glucokinase, form glucose 6-phosphate, acted by phosphoglucose isomerase to give us fructose 6-phosphate, acted by phosphofructokinase to give us fructose 1,6-bisphosphate, broken or cleaved by aldolase enzyme to give us dihydroxyacetone phosphate, glyceride 3-phosphate, which is going to be acted upon by glyceride 3-phosphate dehydrogenase, which is going to add a phosphate group to give us 1,3-bisphosphoglycerate, acted upon by phosphoglycerate kinase to give us 3-phosphoglycerate, acted upon by phosphoglycerate mutase to give us 2-phosphoglycerate, acted upon by enolase enzyme to give us phosphoenol pyruvate, acted upon by pyruvate kinase to give us pyruvate. Pyruvate can go to lactic acid fermentation, alcohol fermentation, or Krebs cycle based on whether there's oxygen or cells. So thank you so much guys, and make sure you keep in touch with me, subscribe to my YouTube channel, like and share so that you can get more knowledge about what happens in our body, especially metabolism. Thank you guys.